Hi, my name is Jody Erber. My mentors on this project are Dr. Eric Gatto and Dr. Sophie George. Our project is titled Upregulation of Heat Stress Genes of Ribbed Mussels in Exposed Areas of the Salt Marsh on Tybee Island, Georgia. The study subject for our project is the ribbed mussel, species Geukensia demissa. Geukensia demissa is a keystone species and a habitat builder of southeastern U.S. salt marshes. This species is invaluable for the health of salt marshes, and it enhances several ecosystem functions, such as water infiltration, substrate stabilization, cordgrass biomass, and species diversity. Intertidal organisms are already at a high baseline stress level, so with climate change causing increasing temperatures, ribbed mussels are at risk of experiencing detrimental heat stress. Ribbed mussels and a species of salt marsh cordgrass, Spartina alterniflora, have a mutually beneficial relationship. Ribbed mussels provide substrate stabilization, and cordgrass provides shade from sun rays for the mussels. Mussels located under a dense patch of cordgrass, which we indicate as less exposed, may experience lower temperatures due to the shading provided by the cordgrass. In comparison, mussels located in areas with very little or no cordgrass, which we indicate as exposed, may experience higher temperatures in comparison. If exposed mussels experience higher temperatures and thereby elevated heat stress levels, their expression of heat stress genes, such as HSP70 and HSC70, may be upregulated. The temperatures of ribbed mussels were collected at our field site located off Old Tybee Island Road on Tybee Island. We measured the temperatures of 10 exposed and 10 less exposed mussels on each of 8 mussel mounds or aggregates. We also collected 1 mussel from each mound for heat stress gene expression analysis. The gill tissues were dissected for analysis and preserved. RNA isolation was performed, followed by an analysis of RNA purity and concentration. The primer sequences for the heat stress genes were found on the NCBI database. The expression of the selected heat stress genes was determined using real-time quantitative PCR. We found that the body temperatures of exposed mussels were higher on average than less exposed mussels. This matched what we expected. The figure pictured shows the temperatures for exposed and less exposed mussels on each individual mound. The letters indicate statistical differences. The RNA analysis determined that the isolated RNA was pure, and the concentrations were also found to range from 105 to 590 nanograms per microliter. The figure here is showing the RNA gel electrophoresis. The brightness of the fluorescent band indicates qualitatively the RNA concentration. So the brighter the band, the higher the RNA concentration. We expected that exposed mussels would have greater expression of heat stress genes than less exposed mussels. Results of heat stress gene expression analyses were inconclusive, though, as many of the RNA transcripts were not expressed. Our finding for mussel temperatures indicated that the shading provided by cordgrass does help to reduce the temperatures of mussels that are located beneath a dense patch. The lack of expression we found could be due to RNA degradation, due to the mussels being left too long in the fridge before preservation. It could also be due to inaccuracy of the mRNA primer sequences, as little information is known about Geukensia demissa's genome. As of right now, no conclusions can be made about whether exposed mussels have increased expression of heat stress genes. In future experiments, the mussels will be dissected and preserved more quickly in order to maintain the integrity of the RNA. In addition, more research will be conducted on Geukensia demissa's genome to increase accuracy of the mRNA gene transcripts. As an alternative test, we will expose the collected mussels to an elevated temperature in a water bath before dissecting them. We hypothesized that since exposed mussels experience higher temperatures than less exposed mussels, that they will be better adapted to elevated temperatures and will have a lower expression of heat stress genes after exposure to a high temperature. Additional tests we plan to perform is an ELISA for measuring the HSP70 protein content of the gill tissues, and an analysis of acetylcholine esterase activity, which could be a potential indicator of thermal stress. Thank you.